Hello. Today we'll be seeing physiology of thyroid hormone secretion. We all know hypothalamus, pituitary and thyroid gland. They are the three important glands which are working in cooperation with each other. So hypothalamus, it produces thyrotropin releasing hormone which stimulates the pituitary gland to produce TSH. And this TSH stimulates thyroid gland to synthesize tetraiodothyronine and triiodothyronine, that is T4 and T3. Now thyroid generates about 100% of T4 and 20% of T3 in circulation. Peripheral convergence of T4 to T3 is responsible for remainder of T3 production. We all know that biological activity of thyroid hormone is mediated predominantly by the intracellular generation of T3 in peripheral sites. So T3 is an active hormone while T4 is a prohormone. So the formation of T4 and T3 is tightly regulated by TSH. Similarly, both T4 and T3 can feedback on the hypothalamus and pituitary to impact on TSH production. In this particular cartoon, what you can see here, the hypothalamic nuclei, they secrete TRH into the portal vessels, and that stimulates the thyrotroph of the anterior pituitary gland to secrete TSH. And this TSH stimulates thyroid gland to release T4 and T3 into the circulation. And this T3, T4, they within the thyrotroph, this thyroid hormone, they decrease the sensitivity of the thyrotroph to the TRH and thereby decreasing TSH secretion. So this is a normal stage of secretion of T4 and T3 with the help of TRH and then TSH. Now let's see how the hormone is synthesis. There are a few steps in the hormone synthesis. First one, iodine supply to the thyroid gland. Second, iodine trapping. Third, organification. Fourth, coupling of this, and fifth, release of thyroid hormones into the circulation. How the iodine is supplied to the thyroid? We all know thyroid gland contains 3 to 20 milligrams of iodine. Out of that, 100% of dietary iodine is the only supply to the thyroid gland. So dietary iodine in the form of iodine or iodide is ingested, and this ingested iodine or iodide, it usually comes from the water, from the seafood, and as a salt. Iodized salt, we all know 30 milligrams of potassium iodide per potassium iodide per kg of salt is available, and 1 gram of salt provides 30 micrograms of iodine, or 10 grams provide 0.3 milligrams of iodide. Iodine is absorbed from the small intestine as an iodide. Iodine supply to thyroid gland is produced mainly, as I said, dietary intake, and that is in the mainly in the form of iodide or in the form of iodine, and that is about 50 micrograms per day. And to maintain the normal thyroid secretion, 150 microgram of iodide intake is must, and this is again, as I said, it is absorbed through the small intestine. Follicle of the thyroid gland is a basic functional unit. The lumen of this follicle is filled with the thyroglobulin, which contains large amount of thyroid hormone molecules. Surrounding to the all this lumen, you will have the follicular cells. They are lined, and so they function as a synthesizer as well as the reliever of the thyroid hormone. Iodide trapping is a second stage in the thyroid hormone synthesis. This is a rate limiting state, and this iodide is concentrated by the thyroid gland, salivary glands, gastric mucosa, placenta, and choroid plexus. Active transport and uptake of iodide by the thyroid gland is taken place with the help of sodium iodine symporter, or there is an iodine pump that concentrate 40 times of iodine. This sodium iodine symporter is stimulated by TSH, and this NIS has an autoregulation mechanism. Other monovalent anions like perchlorate, pertechnate, thiocyanate, they also compete with iodine at NIS. So that's why these are different salts they are used, or we can say these are the different radioactive salts can be used for thyroid scan, the diagnosis of various thyroid. This iodide moved to follicular lumen by protein penetrate, and it binds to tyrosine this is called as organification, mediated by TPO in the presence of hydrogen peroxide, H2O2. This thyroid peroxidase, that is TPO, oxidizes iodide and iodinates tyrosine residues on the thyroglobulin molecule. Now, iodination of tyrosine residues leads to formation of MIT and DIT. There is monoiodothyronine or tyronine and diiodotyrosine. Autoregulation by thyroid gland. If more organification occurs, NIS activity is reduced. And if the organification is reduced, NIS activity is increased. So this is a cycle. Fourth stage of coupling, where nearly 140 tyrosine residues are there in one thyroglobulin. 25% of it is iodinated. 2 DIT comes together to form T4. 1 DIT and 1 MIT together forms T3. A rearrangement of iodinated tyrosine residues of thyroglobulin in the colloid produces MIT, DIT, T4 and T3. These are the thyroid molecules. You can see here the chemical structure of T4 and T3. Then the fifth and important step is release of T4 and T3. Both are synthesized and stored in the thyroglobulin. Proteolysis is the process which is very essential in releasing of these hormones. To liberate these hormones, thyroglobulin is reabsorbed into the follicular cell in the form of colloid droplets which fuse together with the lysosomes to form a phagolysosomes. And then thyroglobulin is hydrolyzed to T4 and T3, which are then secreted into the circulation. MIT and DIT, they are deiodinated by intrathyroidal dehalogenase and iodide recycled. So let's summarize that thyroglobulin is synthesized and discharged into the follicular lumen with the Golgi apparatus and endoplasmic reticulum. The first step is said that ingestion of iodine, aspirated, they are absorbed into the circulation. From there they are trapped as an active uptake of iodide. They are oxidized as an active form of iodine, and then they are entering into the thyroglobulin molecule, thyroid cells, the thyroglobulin molecule. They combine with the thyroglobulin and they form in the follicular lumen. They are attached to the tyrosine in colloid, forming DIT and MIT. In the thyroglobulin colloid, DIT comes together to form T4. MIT and DIT comes together to form T3. This is iodinated tyrosines linking together to form T3 and T4. It's C4. And the step 5, where thyroglobulin colloid is endocytosed and combined with the lysosome, where the lysosomal enzymes, they cleave T4 and T3 from thyroglobulin and diffuse from the follicular cell into the bloodstream to the peripheral tissues. So this is all about the thyroid hormone synthesis in normal state. Thank you.